Hi, this is Dr. Karen Becker, and today I have a very special guest, Dr. Jean Dodds. Thank you, Dr. Dodds, for joining me. Dr. Dodds has agreed to spend a little bit of time today discussing the topic of titers. And titers are a confusing topic. Not only are there different types of titers and different ways that we can titer a pet, when, how, and why to titer become confusing not only for pet owners, but for veterinarians as well. So Dr. Dodds, start by explaining to our listening and reading audience what a titer is.
What a titer is a blood sample where we look at the serum from the blood to determine how much antibody is present to a particular agent, like an infectious agent, distemper, parvovirus, hepatitis, etc. And when we measure it quantitatively, we're actually measuring the combination of that animal's natural exposure to the same disease agent as well as the vaccine history.
And so obviously funguses can cause disease, bacteria can cause disease, viruses cause disease. Talk to me about why some titers, or why some diseases you can catch again, and why some diseases you have lifelong protection, and how this becomes really confusing when it comes to what to titer for and when.
Well, it's even more complicated than that, because years ago when we started doing that, some of the veterinarians that worked for the vaccine manufacturers, for example, said, oh, well, you can do a titer today, and it doesn't mean it's going to be gone. It could be gone tomorrow, and it doesn't mean it lasts at all. Well, that's totally wrong. So what it is certain diseases produce what we call sterile immunity. They would be distemper virus, parvovirus, hepatitis virus in the dog, and panleukopenia, which is sometimes called feline distemper, but that's a misnomer actually. It's a parvovirus in the cat. And when the animal is exposed and recovers, or is vaccinated properly and becomes immunized, remembering that immunization is the effect of a proper vaccination. The act of vaccination doesn't mean it took. Okay, so these animals are immunized and they get sterile immunity. They will have long-lasting immunity measured in their blood through a titer test, a minimum of 7 to 9 years, probably a lifetime. Then we have other vaccinations that do not produce this so-called permanent sterile immunity. And by the way, if an animal has sterile immunity, they cannot become infected again. So they don't get infected or shed the virus should they be re-exposed, because these disease agents are everywhere. Okay, so we have other disease agents that don't do this, like Lyme, like lepto, for example, like bordetella, like the other upper respiratory, kennel cough, etc. viruses, even canine influenza, and they don't last very long. So when we vaccinate for that, it may only last 7 months to a year. And so the antibody levels wane in the blood. Now, does that mean that the animal's not protected? That's unclear. For example, even though leptospirosis may give you a titer after vaccination, that's a relatively low level but lasts for a year, when those animals have been exposed or challenged, at least according to the manufacturers, they are still protected by what we call immune memory cells, which are there for life. In other words, if you have an adverse reaction to strawberries, it's there forever. It doesn't disappear. So the problem, I guess, is how do you know when you do a titer against one of these shorter-lived agents, in terms of duration of immunity, how do you know that they're really protected or not? And you're not going to challenge them to see that.
Right.
So therefore, for those vaccines, if they're needed, now that's a very big if, then we may need to vaccinate more often, like annually or every 1.5 to 2 years, as opposed to the sterile immunity core vaccines, distemper, parvo, hepatitis, and panleukopenia in the cat. Basically never after midlife, once we show that they've got good measurable titer immunity.
It's similar to people who receive polio vaccine, or maybe measles, mumps, rubella, that protected for life, versus, let's say, tetanus, where because it being a bacterium, oftentimes MDs will recommend that a person has an additional tetanus shot because immunity may not be lifelong. Is that a good correlation for the average person to think about?
That's partly, yes, generally. But remember now, we've had some recrudescence or reappearance in elderly people for polio and measles recently. So there's been some question now whether it is lifelong or just decades long. And of course, in the defense of those vaccines, they're not made the same way today. We have much better vaccines with more properly made, because the technology wasn't where it is today 30 years ago.
So with Bactrins, which are vaccines made for infectious agents that are bacteria, because it's hard to trick the body into actually having consistent protection, talk to me about the risk of, let's say, a Bactrin-based vaccine versus a modified live vaccine that would be given for a viral infection. What are your concerns pertaining to those two different ways that the body can become infected with two totally different organisms?
Well, that's very complicated also in a way, immunologically, but let's try to put it down into common sense. Bacterins generally are not modified live. Obviously, they don't replicate in the body. They don't multiply, but they're also, you could call them toxic tissue culture soup, munched up stuff. Like think of the rattlesnake vaccine, for example. I mean, it's not a highly purified stuff, so it may not last very long, but it's also not as purified as the modified live vaccines. So why did we go to modified live vaccines, except for rabies, from killed vaccines years ago? Because they produce a more rapid, more complete immunity. I was one of those people that kicked and screamed for years and said, kill parvovirus vaccine, even though it doesn't last as long and may not mount the immunity as quickly, is actually safer. And that's what the late Jonas Salk said. Killed vaccines are always safer than modified live if you have them and they work. So the modified live vaccine produces complete immunity. It challenges your secretions, your secretory immunity, your cell-mediated immunity in your tissues, and this circulating immunity in your blood at the same time, and relatively quickly. I mean, distemper vaccine produces immunity immediately. For example, an animal you don't know has any protection. They're exposed to wildlife distemper virus. If you vaccinate them at the same day, they'll start producing antibodies right away and should be protected.
Wow.
That's amazing.
It is amazing. And how about parvo?
Parvo, no. Parvo's a little different, and parvo, of course, is much more prevalent, especially the new virulent 2C variety. If you're exposed to parvo the same time, the animal will get sick, not as sick. So it's vaccinated, but it won't start to produce antibodies for maybe 72 hours or more. Wow. So they get partially ill. So parvo is not the same, and it's much more prevalent than distemper is, unless you're running around feral animals or with wildlife.
Okay, so we've established that we've got Bactrin-based vaccines. Just on a side note, briefly, talk to me about Kennecough vaccine and why that isn't necessarily there's intranasal and injectable, but why animals can receive vaccine and then in turn be immediately infected with kennel cough. Talk to us about why that vaccine isn't necessarily a panacea for anything.
Well, the upper respiratory virus vaccines are not 100% effective. I just got an email where they claim they're about 70% effective. But we have people that take animals in for foster, for example, do boarding in their home or boarding and grooming kennels that are really worried about the possibility of their liability and even minor risk. And so this one client, well-meaning, wants to do Bordetella every 6 months. If you do that, it should be intranasal.
Okay. And so what are your thoughts on injectable Bordetella? Do you recommend it ever?
Well, I wouldn't, because the intranasal Bordetella contains interferon, and interferon also gives you cross protection against the other upper respiratory viruses. Not completely, but so everything is partial. So parainfluenza, canine influenza, adenovirus 2 for kennel cough. If you give the injectable Bordetella vaccine, it doesn't contain interferon, it doesn't do that. And you cannot give an intranasal injectably or the injectable intranasally. Somebody did that recently to a Rottweiler and it got acute hemolytic anemia.
Oh my goodness.
Well-meaning,
Sure, my goodness. Okay, so let's back on the topic of titers. Very confusing because there's so many different ways to titer. Let's just start with hemagglutination, because that's one of the ways, depending on, people nationwide recognize, maybe my pet doesn't need annual vaccination, maybe the pet wasn't titered early on and they're thinking about doing this now. They go to their traditional veterinarian who has no idea even what they're speaking about, but their traditional vet loves them and wants to work with them and is willing to do anything. So the traditional vet calls the local laboratory and says, listen, I have a client requesting a titer. I don't know what's going on. Do you do them? The lab says sure. The veterinarian probably doesn't even know what to ask pertaining to what type of titer they're running. So let's just start with hemagglutination, because HA is a common way that some labs are performing titers. What is hemagglutination and how and why is that run?
That's the gold standard, for example, for measuring parvovirus immunity. It's a very, very old technique. It's been supplanted mostly today with the more efficient ELISA, enzyme-linked immunosorbent assays on serum. They're faster to do. There's very good ones available. Now, the parallel to HA is serum neutralization, and we use that technique as has been the gold standard for canine distemper virus. But again, ELISA techniques have supplanted that for most places because it's too cumbersome, and so therefore it's going to be more costly if you have another method that works well.
So, because ELISA has basically replaced HA and serum virus neutralization, talk to me about IFA or immunofluorescent antibody technology, and where does that come in?
Well, the immunofluorescent IFA assay is something I'm actually quite proud about because many, many years ago now, it seems, in the late 1990s, I helped develop such a test and published it in the refereed literature with a senior veterinary extern, Dr. Lisa Twark. In 2000, we published a huge study in about 1,500 animals, and we compared HA and SN to the new IFA test. Now, why is it an interesting test? And I like it a lot because you're actually doing what you do when you diagnose a disease for those same agents, where you layer the sample from the patient over cells that are infected with the virus agent, and they fluoresce under a microscope. They're just shiny fluorescent green. You layer the serum from either a presumed clinical case or an animal that's been immunized over that, and you see if it dampens the fluorescence, and then you quantitate the difference. Now, vaccine titer levels are going to be much lower than disease levels. So you're going to start dilution of the patient's sample much lower than you would if you were looking for a disease agent, because you don't want to waste the cells. It's too expensive. I like the test because it's actually simulating what happens to a natural infection, seeing how well the animal is protected with that same methodology, but it's more cumbersome, of course, and it's offered by one of the major clinical diagnostic laboratories.
So, and I like IFA because in essence, it's, when I say a yes or no answer, we're not actually looking for an exact number of antibodies. We're not counting antibodies. We're really looking for yes, you have a response, or no, you don't have a response, which I think for a lot of owners, it helps reduce the stress of, is my dog protected or is my dog not protected? Do you agree with that?
Well, that's true, but there's one caveat. The standard of whether you get a yes or no answer for this particular laboratory was set based on the studies we did, not at a minimum level, but at a moderate or higher level. So if you get a yes answer, that means the animal is well protected.
Which is great. So if you're going to err on the side of being overprotected. If the animal is not protected via IFA, they're simply not protected. You would need to revaccinate to provide immunization. Is that correct?
Only if the animal isn't genetically unable to do that.
Right.
And we mustn't forget about the genetic non-responders or low responders. They'll be that way for life. And no matter how many times you vaccinate them, they will never mount immunity that's protective.
So let's talk about the fact that some labs run ELISA, and in my own practice, we've checked via ELISA, and the animal does not have notable protection against distemper, but then we can recheck with IFA, and there is protection. This is a quandary and confusion. Can you help shine some light on what could be going on here?
Well, at Hemopet, we do our own in-house laboratory titer testing, and we use an ELISA technology, I must tell you. We have recently evaluated a second ELISA technology, and what we do, if we get a negative, and it's nearly always to distemper, like you said, and these are not non-responders, these are animals that have had measurable titers before, for example, we always send it out for the IFA confirmation test. Whatever results we get, we give both sets of results to the veterinarian, so they can help make a professional judgment decision about whether one is negative, the other is positive, because they are probably measuring slightly different reactive antibodies on the surface of the cells and the antibodies in the serum. So now, for distemper, I'm not so worried unless they're around wildlife. For parvo, I would be, because it's parvo 2C. So let's say you got a negative on parvo with an ELISA, which is unusual, by the way, if you use the best technology. And there are many different labs who are doing it, but we believe the one we use is the commercial one is the best, okay? And it's been validated by others. So we do that. Let's say it's negative. We send it to the IFA lab, and it becomes positive. It's then going to be the veterinarian's judgment, given an endemic, epidemic, or whatever, whether a single parvovirus vaccine booster should be given, as opposed to, well, I only have the combo wombo in my fridge, so I'll do that. We don't want that.
Sure. And that also provides additional confusion, because if you do have a questionable result, and most veterinarians are running these tests not out of a history of being able to provide lots of background, they're doing it because the client's requesting it and they're trying to be compliant, and we respect that. The frustration is they don't know what to do if they're with some of these challenging cases. So single agent vaccines, of course, are significantly less stressful to the body immunologically. But talk to me a little bit about, in those cases where you have potentially maternal antibody, talk to us about where there could be some coverage and some confusion pertaining to maternal antibody. In fact, let's go back and talk about what is maternal antibody and what happens during this window of opportunity, which is why vaccine is even instituted. So maternal antibody.
Maternal antibody is what the mother gave to the puppies, partly in the placental circulation during fetal development, about maybe 10-15%. The rest of it comes from the colostrum. And so when the puppies or kittens suckle from the mother, they get antibodies. And how long those last is going to vary with how high the mother's antibody titer was when she was carrying the babies. They usually are gone by 16 weeks. But they start to wane around 8, 9, or 10 weeks. And so we have this, as you say, window of vulnerability between, say, 10 weeks and 16 weeks when the animals are not completely protected. So what do? We vaccinate earlier. What that does is temporarily suppresses the maternal immunity that's there. So the animals become more vulnerable for those few days, 3 days, 5 days, 7 days after the vaccination at 7 weeks or 10 weeks, for example, when they're also the maternal antibodies waning. So we sort of got a catch-22. So it's important the animals not be out and about in the dog park or bark park or whatever park, that they be kept socialized at home or with people they know or with neighbors or in their backyard, not the front yard, because you don't know who's been around the front yard, to make sure that they're well immunized before they go out in the real world.
And then titering 2 to 4 weeks after that last puppy shot would potentially be a good opportunity to determine if those vaccines actually immunize the patient, correct?
That's correct, but they should not be done before 16 weeks, because if you measure it earlier, and many breeders do that, they're not aware, you could be measuring maternal immunity, which is going to be gone.
Right.
But there is one other thing about titers, and that is, I believe from our parallel data studies, and we do thousands of these tests every year, that the IFA test will still detect some maternal immunity a little bit longer than the ELISA test will. And I'm not sure why that is, but possibly because we're using different technologies, as I mentioned before.
Okay, all great information. So let's talk a little bit about rabies vaccine. Rabies vaccine, depending on your state, is approved for dogs as young as 3 months, in some situations 4 months, and then holistic veterinarians will oftentimes try and push that vaccine off to 6 months or so to give the body a chance to be a little bit more developed, because that's a hard vaccine in terms of reactivity. Talk to us a little bit about duration of immunity for rabies, because it is a killed product.
Yeah, well, part of the problem is people still think that we have modified live rabies vaccines. I was teaching last weekend and somebody said, oh, but they're horrible. The animals could get rabies. I said, well, we did years ago have modified live cat rabies vaccines, and they did occasionally produce rabies. There are no modified live rabies vaccines. People should know that. It's sort of like a Bactrim, but it's a killed virus product. Very, very potent. We don't want to give it earlier than we can. Even though the label says you could give it at 12 weeks, we don't want to do that. We don't want to give it before 16 weeks. The California legislation recently was in a big controversy about that, but we've got that resolved, thankfully. So how long do rabies antibodies persist? Well, the antibodies in the blood through titer testing can be 7, 9 years, maybe longer. The question is, are those animals truly protected? So the rabieschallengeFund.org has just finished the 5-year of the 5 and 7 year trials. We're about nearly 6 years into it now. And with the studies done by Dr. Ronald Schultz, the animals are rented actually, so they're in fine, lovely kennel home situation. We've actually done a study on them, and we've looked at the antibody titers all the way through, 3 years, 4 years, 5 years, etc. 3 years, everybody had good immunity. 4 years, they had measurable immunity, but less. 5 years, some of the animals did not have measurable titers. However, we found out with a vaccinal challenge, not live virus challenge now, that they produced good immune memory cells. They all responded extremely well upon challenge, even though some of them had titers below the 0.1 international units per mil, deemed to be adequate to protect a person against rabies. So we're confident that even at 5 years out, that these animals have good immune memory to protect them from rabies upon exposure.
Isn't that neat how the body works, where even though it's below that standard level that we all accept as being adequate, the body still is capable of having this anamnestic memory response to where the body knows what to do, even if it's not a detectable level, the body knows what to do, which is neat. It's great that we're beginning to give some credit to what the body innately knows what to do.
Wasn't God great?
Yeah, honestly, I mean, just amazing. It really is. And so it's kind of sad we have to prove this, but yes.
I just wanted to clarify that even though we know that the immune memory response was boosted in all of these animals, that does not mean that the USDA would accept extending the licensure yet. And we haven't got to that stage.
Sure.
We're just developing data that nobody's had before on the actual titers in dogs, as opposed to extrapolating from people.
And wonderful, this is the first study of its kind. Takes a lot of money, right, Dr. Dodds, to do this? You have another year of research, and not only in doing the research with Dr. Schultz, then you have to collect the research, publish the research. It takes a lot of money.
That's correct.
So we are partnering with Rabies Challenge Fund to help raise the additional funds necessary to not only complete the study, but to make sure that it's published in a way that provides everyone the most information that's going to benefit the most pets.
Right. Now, on the rabieschallengefund.org website, there's not very much written about what I've just told you. And the reason for that is we can't say too much, otherwise it precludes a refereed publication.
Sure.
It would be considered prior publication, then it's not novel. So we have to be sort of a little bit careful about what we say on the website.
Sure. But you're still obviously actively doing the research. You have about a year left, is that right? You're just
correct.
6 years in?
Yes, we're collecting money. HemoPet monitors and manages the fund and the money from our office in Southern California. None of the people are paid by the Rabies Challenge Fund, nor is Dr. Schultz or myself or Christine. Nobody receives any salaries whatsoever. Only the care of the animals is involved. And my staff at HemoPet and the time involved is donated by us at HemoPet to support this study.
We appreciate you not only doing that, Dr. Dodds, we appreciate everything that you do, and this is your opportunity to be able to help support this research, this much needed research, and we appreciate being able to partner with Rabies Challenge Fund, Dr. Dodds, Christine, as well as Dr. Schultz, to be able to provide this opportunity to help all of our pets live a better healthier life by potentially extending the vaccine frequency that will be required through this really important research. Thank you for joining us, Dr. Dodds, and thank you for the information that you provided to us.
Thank you.
Mercola Healthy Pets is happy and proud to be able to partner with Rabies Challenge Fund in a fundraiser that can help improve the lives and well-being of the animals that we all love. During this week, for every dollar that you donate to Rabies Challenge Fund, Mercola Healthy Pets will donate $1 and match the contribution that you give.